mm, mm, mm. Hello.
Hi, everybody.
Hello.
Hello.
Hi.
This is my first time here, and I'm new to the group, so I don't know if you all do introductions or anything like that, but be happy to introduce myself.
Great, we should do that. We haven't done that for a while. Let's do that. Hung, you're chairing. Is it you said chairing? I'm sorry, I forgot.
Should be me this time, yes.
Okay. And I think I'm minute taking, which is good because I've got builders working downstairs, so the noise is not good.
Okay. I guess I'll just start with myself and then we'll just go clockwise from where I can see everyone. All right, so yeah, hi, Wally. My name is Hong and I am from the University of Sydney. I am currently almost to the end of my PhD. So I'm doing mycetoma, obviously, which is why we're here. And I'm also doing a little bit of chemistry educational research as well, where I look at research and outreach activities, experience of students and things. But yeah, I'm here today, as always, just to show some support for mycetoma project and also to do other cool stuff here. So I guess Wally, this is you're next on my screen actually. So would you like to just share with us a bit about yourself?
Sure. So my name is Walid Osman. As I mentioned, this is my first time joining the group. I'm actually in the third semester of my PhD, so I'm just at George Mason University in Virginia. I'm just formulating my dissertation, and I would really like to focus on mycetoma. I have great interest in mycetoma because I am from Sudan, and it is endemic in the country that I'm from. I've been doing a literature review on mycetoma and came across MycetOS. I am also a senior bioinformatic engineer at a small therapeutic company, the drug discovery company in California called Interline Therapeutics, where we focus on developing drugs to target protein communities. So our main technology is APMS, and we would like to characterize protein communities using AI, structural prediction, and elucidating how to change a protein community from its disease state back to a normal state. We're currently focused on developing small molecules and E3 ligase for protein degradation.
Yeah, thank you so much. Matt, you're next on the screen.
Hello there. I'm Matt Todd. I'm a professor of drug discovery at University College in London. My lab does organic chemistry and a little bit of biology. I was one of the people, along with others on this call, and along with D&DI, responsible for setting up MycetOS back in the day as an open source way of developing new potential medicines for mycetoma. Nice to meet you, Wally.
Likewise.
Bernard, you're next on my screen.
Yes, hello, I'm Bernhard Piersack from University of Bayreuth in Germany. I'm chemist and joined the group last year to achieve some new drugs against mycetoma from our own lab here in Germany. And yeah, that's it. Thanks.
Yeah, thank you. Wendy, you're next.
Hi, I'm Wendy van der Sande. I'm an associate professor at Erasmus MC in Rotterdam. I've been working on mycetoma for more than 20 years now, and I'm responsible for the drug screening. So I'm the biologist in the team. So we do most of the in vitro screenings. We have the in vivo Galeria models over here. So that's our part of the job.
Yeah, thanks, Wendy. Ma, you're next.
Yeah. Hi. My name is Yingyi Ma. You can call me Ma. I'm a PhD candidate in Erasmus Medical Center in Rotterdam. I'm in my third year for my PhD, and I work with the in vitro susceptibility test and in vivo test in Larvae. So nice to meet you.
Yeah, thanks, Ma. Yeah, I think we also have some other collaborators who are not always kind of like online. And so one of them is Mandrake, who's not here today, but he's kind of like jump in and out from time to time. I think there's also Dimitri, who works with Matt at UCL and also a PhD students, and we have various other organizations as well. Also from UCIT, there are 2 new honor students who will be working on mycetoma as well. So I think so they're not quite ready yet. They're still on sort of like the induction stages, but I guess once they're ready, I'll also get them to say hi, maybe like post something on GitHub to introduce themselves and what they like to do. So most likely it would be something like a series 2 or series 5. So just something that they're keen on getting their hands on, getting some experience as well. But I'll get them to say hi in probably a couple of weeks' time.
Yeah, if they could obviously sign up for GitHub, that would be great, because then we can communicate with them. And Walid, if you could also sign up for GitHub if you're not already, then we can include you on the various things we submit and talk about online.
Absolutely. When you say sign up for GitHub, I mean, I certainly I've had a username for quite some time, but do you mean to, can I add myself as a participant to the repository, or?
If you send me your user ID, I can add you to it, so to make sure you're included.
Absolutely. I'll do that now. Thank you.
Yeah, the 2 new honor students, I think they might also be using GitHub as their open lab notebook. So it's something that Clementine's been developing. So yeah, they'll definitely have a GitHub, and I'll just let them know to join into mycetoma as well when they're ready. All right, so I guess we'll just moving on to the agenda that we have for today. So that should be on this link here. If anyone needs it, that should be here. All right, so the first one is actually going to be on the suggested compounds that we need to get from the Epichem library to be sent for a request for shipping. So I guess I had a look at the list again, and I guess I can share some of my thoughts. So let me just share my screen. Give me 1 second. I can. All right, so here we go. Okay, so I initially sent this one, so I guess that's just missing. So it's just missing context essentially, which Matt has pointed out, and I think Dimitri also added a few more compounds as well. So initially, there's a really large set of compounds that we then needs to narrow that a little bit. So I guess I'm going to talk a little bit more about this one right here. So I actually organize it together in a sort of more cohesive way, which is this here. And I will just explain this in a moment. So basically, there's sort of like, this is like a mind map that I'm making just to show why is that I would like some of these to be shipped to Wendy for testing. So anything that's like in an oval shape is all of the current SIR that we have already for phenomenal. So like we've got 1, 2, 3, and 4 here. And then anything that's like in a square is things that I would like to have as a suggestion for Martin or whoever's from the team, from the EpiChem team to ship to us. And yeah, these are just like the directions, like the thoughts process and the links. So they're not actually reaction, they're just to show a flow essentially. So say the first, so like the idea as well is because the pool of compounds from the EpiChem library, because it was tested for something else, it was quite diverse, which also means that it's a little bit hard to match pair all the time, and especially to match pair with what we already have in tested as well for SIR. So what I thought is that because some of them we cannot directly match pair, we can actually so like have a point or a compounds where we can match those two, and then those one can then again match to the to one of the target, for example. Say for example, like these one won't be directly matching to this, but like through here we can sort of like make an inference as well. So it's sort of like a secondary level of matching. So that's what this means. And I guess the aim as well, when we're asking, as Matt had mentioned, that we should also be mindful of the effort it takes for people to find these. So we just need to be efficient, and it just need to have a clear reason. So that's why I just want to limit it down a little bit. I believe from our November, it was suggested that we should be around about 10-ish compound. So I got about 13 here as a final suggestion. So for example, what we currently have here is these two that we know, the in vitro and in vivo result, and I think these are actually quite good. These are one of these are our initial lead actually. So we can get something like this compound here. What that does is it allows us to then also have a look at this one where we just change one thing, which is the substitution pattern on this 3 pyridyl ring right here. And that would hopefully show some more so like exploration of variation here. And then there's also one of this compound which I found really interesting, is that it has a 2 substitution on both of these. And we found that, or what I have written in the manuscript draft itself, is that the 2 substitution is quite important, as we found out in the SIR. So, but we don't really have one where it's 2 substitution on both sides. And so this could be something that is quite interesting. And whereas this one right here, because this is exactly the same, except for this change right here. So what we had in the manuscript right now is that we know that 2 substitution is quite important, but 4, we're not quite sure yet. So there's an avenue to explore a bit further. So this is one way that I would suggest we do that. So first, this is by removal, and it's also match pair as well. And then we can compare that potentially to this one, and then it goes back to the target. And then move on to this. This
so one. Just quickly on that. So the issue here. So the compounds in the top left.
Yeah,
they are both active, right?
Yes.
And so the only risk here is that if the compound immediately below the oval, so the one ending in 027,
yeah,
the only danger here is that that's inactive. And then if all the others were inactive, then we don't learn anything. But I guess the point about that compound is we hope it's active because it's this pretty small change. And the activity of that would allow us to compare the ones beneath it. So that's fine. In all cases here, we just need to be clear that the existing compound's active.
Yep. So basically, like the selection here allows us to pinpoint in terms of like really close to a match pair where like just one change and everything else is kept the same. So that's why I suggested these. And then for this current active, there's 3 of these right here as well, which are I believe most of these are at least active towards like at least MIC. One of these is the lead compound as well, the initial lead as well. And then there's 3 areas of match pairing changes that I can see right now. So the first one is because we also say that the 2 substitution is important, which means that I can then have a look at how that will be changed. So things from a fluoro to just a methoxy, for example, or then we have a fluoro, but then we also have another one here at the 3 position. So that's why I suggested that there. And then this one here is also linked to that one as well. So the only change here is again the 4 substitution. So again, that's also a match pair back to here, which also then open us to explore relationship between these with that one as well. And then the other change here is a 3 substitution on this side. So from a 4 halogenated to a 3 and to a 3 CF3 here as well. So one of the thing that was lacking in the current SAR is exploration of 3 substitutions. So that's why I picked these 2 as well as that one right there as a point of to explore further. And this last one here is one of the only one that I think this one either myself or Dimitri found that has, instead of a 3 period, it's a 2 period. So it's a change from that to that one there. And it's also a match, it would be a match pair to myos 002 as well, with the only change being that position right there. So that would also be quite an interesting compound to look at acquiring for testing. So I guess those are my original suggestion, I think. And then Dimitri also had a look again in January and then suggested 1, 2, 3, 4, 5, 6, 7 extra one. And I guess some of these are quite interesting. And the only reason that I would say that I sort of like excluded some of them out initially was because some of them we already kind of have a representation already. For example, like this one right here, we already have one of like another halide here already instead of that one. So I think it's quite interesting if like numbers and are not like I would include this if it's so like we can just ask for more. But I'm also mindful as well that we kind of need to be, not ask for too much of a favor as it already is. I know that these 2, I had a look, these one we've tested multiple times already, so I don't think we should be asking for more of this. These are actually myos 3 and 20. So we've already done this testing. So let's just not ask again. These 2 are interesting, but again, they kind of fall into that's like, it would be nice to have, but like we don't really need to have it right now. Because one of the things that was lacking as well, I think, was by substitution on one ring. And this is the one that will kind of like answer that. And it will go quite well with, because we already have this one here. It will also be nice to also have this one. But I don't know. This one might probably be nice to have as well if we have the capacity to ask for them for this. What do you think of these 2?
Yeah, I mean, it just depends on the idea behind some of those compounds was to test the 3 substitution and the di substitution.
Yeah.
So I think the ones above are the slightly more important ones, and the ones below are the nicer to have.
Yeah, that's my thought as well.
But the bottom left ones with the CF3 groups there.
Is it this one or this one?
Yeah, so the 2 that have been tested already.
Yeah, this one.
So do you know if they're active?
Oh,
because that answers some of the CF3 concern I had previously. That if you go up to that sort of bridging compound that you had, that I think that's the one with the CF3. So if those are active, it would suggest that that compound's going to be active, which is good for us.
So A4. Oh, for CF3. I'm not sure. I have to go and double check because I don't have it right here.
Okay. Just adds weight to the idea of that bridge compound, because the one at the top of your screen, top
So
left.
this one right here.
Yeah. So that had the chlorine. Okay. All right. So the ones on but just below that on the left, hopefully then the compounds of the CF3 would the CF3 helps then that hopefully if the existing ones are active.
Yeah, so your point is just to come back and check the activity of these to see how the 4 CF3 contributes instead of like activity in general.
Yeah, I mean, if all those CF3 ones are inactive, then it just maybe we need to reevaluate whether we're sending the ones in the box above that.
Okay, yeah, that makes sense. Because the only thing is the thing with these ones as well is these are the only one that I can find that kind of like there's only one chain to it. So again, that's just it's just what it is, because like we didn't make this, so we just gotta have what we have. But yes, I see your point.
Yeah.
Same for me. I couldn't find any additional ones for the CF3 motive.
Yeah, this one right here, I think Dimitri also suggested this one. I found already one that we have currently in the SAR. So this one is not a CF3, this one is just a chloro on this position right here. It matched exactly to this one, but however, this one right here doesn't really work quite well. The in vitro is not quite strong. So I would say, again, it's not as high priority as what I would suggest for like this one right here. So I guess these 3 would be highest priority, and then this one, and then this one here. So I would say not to include this right now.
Yeah, if the comparator is inactive, then yeah.
And then this last one right here is this one here, where the change is a alcohol to a fluoro, and then that match to a batch that we already have here. So these 4 we both have in vitro and in vivo for this one right here. I'm not quite sure, like, what do you think about this one as a comparison to these 4? Because to me, it doesn't jump out as something like super interesting, but I could be wrong.
Are those the closest compounds? So we don't have the same, the same.
We do have one that is a chloro 2 here, so it's a MyOS 2 right here.
Yeah, so if that's active, then
so this one right here. So it's active all the way up to 50, but then the in vivo is not quite significant.
Okay, but I think if it's active in vitro, then that would allow us to determine whether the OH could be replaced by an F.
Okay, so this one's quite good.
Yeah, provided it's a match pair and the original is active in vitro, then we can compare.
Yeah, it is a match pair, and it is the original is the one that we compare to is actually has good in vitro. Yes.
Okay, then it's worth doing, right?
Yeah,
because the middle replacement we've got amine, methoxy, and H.
And also, we're making a point that the OH is quite important. In the general paper, like R&D section in the discussion, that the OH hydroxy group is quite important for potency. So because of the hydrogen bonding, I think we're making this point. So I thought it would be quite important to get this one with the CF, the central carbon atom, tested as one of the priorities as well.
Can you zoom in on the top right?
Yep.
So the amine and the methoxy were, so the methoxy is just about as good, is it?
So this one, so top row is in vitro, bottom row is in vivo. So I think the alcohol is the best, and then followed by smaller are better. So these 2 are really good in vitro, and then followed by the amine, and then the H. If we remove everything, then it's bad totally. And then if we go back to in vivo, then hydroxy is best, then amine, then methoxy.
Right. So I mean, in vitro, the data suggests that the OH is not important, right? If the methoxy is just as good.
Yes.
So the flora would be a test of something that mimics the H, because it's small but electronegative. So it would be useful to know that. If it's also potent in vitro, then that tells us something.
Okay, yeah, that makes sense. Yes, Walid, I think you raised your hand.
Yeah, I had a number of questions. First, can I ask, with this series of compounds, what the target is and what the mechanism of action is, and whether you envision that being able to overcome the encapsulation of the mycetoma lesion?
I guess, Wendy, would you like to talk a bit more about this point?
So this one seemed, well, we hypothesize it's a CYP51 target like the azoles because, well, the known compounds do have this as a target. We did look at in vitro susceptibility first. So that's what you see with the MIC50 values on top. But to see if it also penetrates the grain, we use the larvae model in which we can form mycetoma grains, and those grains are encapsulated as well. So if they enhance larvae survival, they must have been able to penetrate inside the mycetoma grain, because we started treatment at the time point when the mycetoma grain is already formed. So they should have been able to penetrate inside the grain. And that's actually one of the things we are testing here. So how can we modify the molecule so that it can penetrate inside the mycetoma grain and also inside the Madurella cell, of course, to perform its activity?
So you said it's a CYP51 target, SIP?
Yeah, so as the azoles, so itraconazole, ravuconazole, posaconazole.
Okay.
And also we're aiming for ergosterol biosynthesis inhibition, which is essential for the cell membrane of fungi.
Okay.
Also, Wendy, before I forget, there was a paper I saw, I can post it on GitHub, where they did a proof of concept studies where in the biological assay, they measured the levels of different precursors of ergosterol. And that's how they have proven that it's actually the CYP51 pathway. That was one of the methods. And I was wondering, have we done something like this in our studies or not? So there is a chain of methylated precursors leading to ergosterol, and they measured them by using chromatography. I think something like this. It's like should not be a very difficult method, but time consuming probably. And that's how they proven, like by measuring different percentages of those precursors and ergosterol, the final product as well.
Yeah, I know they do that also for yeasts. They had a paper where there was a multi-resistant isolate, and they proved by ergosterol measuring the different components of the cell membrane, actually that the different mutations linked to different types of ergosterol in the cell, well, the sterile compounds. But I don't have the equipment here, so I cannot do it in my own lab. Maybe somewhere in the university itself they will have it, but I didn't do it myself. Also not sure if I can isolate the ergosterol directly from the grains. I'm sure I can do it from the fungi. It should not be different than from a yeast, but the grain, it's really, really difficult to destroy the grain. So it took me already a while to get DNA out, let alone something else. So of course, if somebody online is able to do it, I'm more than happy to send the samples, but yeah, I myself, I don't have the machinery here.
I just had one follow-up question. What bioinformatic challenges or efforts are being conducted? Are you all, say, for instance, calculating the relative binding affinity of these compounds with the target?
Well, we haven't done that yet. The genome of Madurella is available, but all the gene predictions are made in silico by comparison. So we don't have crystal structures. We only compare them to crystal structures from other species, for instance, Aspergillus, if it's available. So we didn't do that yet. We didn't have somebody with the expertise. So if you could contribute to that, that would be great, of course.
Yeah, I would love to. We've shown in experiments that we've conducted that AlphaFold's prediction value is on par with almost as good as some of the crystal structures that are elucidated in through experimentation.
Yeah, I mean the AlphaFold CYP51 should be fine, right? Because it's a machine learning model, so it's taking what's known and predicting what's the structure of what's not known. But if the protein is part of a family of proteins which are quite conserved, and the prediction is going to be very good, I think. So the ones that are already on AlphaFold, I would expect to be pretty accurate, particularly in the middle of the protein.
Yeah, I'll certainly see what I can put together. I think doing the structural prediction and maybe even doing some prediction of multimers, or trying to determine other proteins that could bind to CYP51 and determine if you know, if there is a relative binding affinity and how strong that is, could be another avenue of to pursue.
Yeah, we have a group of students who from Cameroon actually, who want to try and do some modeling of compounds into the relevant target protein. And if there was something we could do in terms of providing either the AlphaFold prediction or something which is combined with the AlphaFold prediction into just a PDB file of the protein that we could use as a kind of docking target, that would be helpful for them, I think, because then they could start to play around with molecules and try and rationalize some of the SAR we're seeing. Anything of that kind would be great.
Absolutely. I'll follow up with you about that.
And also maybe for some of the other series, there we're still a little bit blind to what the target should be. So,
well, I think for benzimidae cells, it's quite clear that it should be tubulin as a possible target for these series 5 compounds, right?
yes, but I'm not sure if we figured out what the series 2 binds to. But yeah, we could do a similar thing in series 5 as well, of course. But at least all the in vitro data are there. So in case anything else is needed, please let me know.
Okay, that sounds great. I guess we'll just move on to the next few points. So no, we don't run out of time. Next one is actually the manuscript. There's a few more points. So I just had a look at the manuscript before, and I just had it again. I think in the experiment section, there's just a few, 1 or 2 issue with referencing. That just need to be fixed. But I guess, is that something that we should, because like I'm also aware, like I use the Tarot, and I think someone else is using EndNotes, so it might be a little bit of a problem. So it needs someone to do the collaging, I guess. Yeah, I'm not sure how, any ideas on how we should be doing this.
I mean, I think the referencing is not so tricky. It's there are just still quite a few comments from Kimberly and other places where data are needed or formatting needs to be changed, that kind of thing, in the main manuscript and in the separate experimental file for the chemistry. They just need so those 2 bits just need to be made ready for submission. That's all. I mean, I think neither of them are quite there yet, so they just need to be tidied up, comments removed, any red text filled in. any remaining data filled in, those things.
Yeah, and I think the references should be done at the final part when the manuscript is just ready to submit, because if it goes back and forth and people still add references, you need to do that over and over again. So I would recommend only to do that just before submission.
Yeah, so same thing as when I wrote the SAR, I have a separate file where all of the references live, and I can just hand it off to whoever is doing the final referencing. So that's currently not in the manuscript yet, but I've got that separately ready for that section as well. And I think I'd also have that for my experimental section, so that should be ready. So I guess it's back to, I believe that will be me and Dimitri to have a look at that, and I'll let's just.
So I had a look at the manuscripts just before this meeting the previous week, and there's one problem which I found like is very hard to resolve. I think the better solution would be for me, Han, and Kimberly, ideally if she's still in the project, to meet up like separately in a Zoom meeting and just to tidy up all the comments, because I found personally impossible to do this. Like I cannot do comments, the ones that Han did, for example, the parts. We have the application of general methods. There's a bunch of stuff that like I cannot start changing them only on my own.
Okay.
We need the other side to know of those changes as well. So I think maybe a short meeting would be much better, because we really need to speed up on this.
Okay, so how about let's meet next week. I'll have a look at the manuscript again sometime this week, and we can come
because I also added some additional comments that were like from your that were related like to your part as well.
Okay, yeah. I'm not sure.
Our
Kimberly...
different references, applications, general methods, not major things, but it's a bit hard to decide on this on just for one person. So it's better if we can meet, I guess.
Yeah. I do have a general idea what Kimberly wants as well. She might not be available immediately, so that might take some time. So let's, why don't you and I just meet together next week to work on that, maybe like half an hour to an hour to deal with it. I'll read again all of the comments sometime this week, and we'll just find a time. I'll post it up on GitHub as an issue, and then we can work out a time next week, maybe. Yeah.
Sure.
Okay, yeah, that's, so we'll just leave.
So the plan is we kind of finish this off SAP, and then we let Matt know that he can have a look at the final draft version, which is one of the objectives on the GitHub issues at the moment.
Okay. The next one is manuscript again, so supplemental file. I have downloaded my my notebook, my lab archive notebook. So is that as a PDF, or do we actually want it as something else? Because there's another version where, say, it creates a different thing.
It's very much not the PDF, yeah, because when you PDF it, you ruin the data, so you can't access the files anymore. So it's the offline copy of the
offline copy of the whole lab notebook. I'll do that.
Yeah. If you need to distill out something, or if you need to slim down what's being shared, for example, if you have other things in the lab notebook and you want to tidy it up just to the relevant things, all the experiments, that you've done for the project, but sometimes people have other things in the lab notebooks, then you can just copy that over to another lab notebook pretty quickly, and then just create an offline copy of that. And then you download it, and then you upload it somewhere which is available, like, UCID's e-repository, and you get a little URL, which is the link, and then anybody can see it.
Yeah, I did have that problem where, like, there's a big section of it that is shared, but then there's also the bits that I don't want out because it has nothing to do with the project, but it's just like my personal notes. So I did have that problem when I tried to create an offline copy, and then it has everything. So
you just copy it. So create a new lab notebook with a title, like manuscript submission lab notebook or whatever, and copy everything you want over to that, and then zip that.
Okay, okay, sounds good. I'll retry that again, and I'll post that version. I'll drop it in the Dropbox actually, where everything for the manuscript is.
It may be huge. So if it's
Oh.
huge, then just
Wait.
put it on the e-repository because it may be hundreds of megabytes.
Okay, okay, yeah, sounds good. That's second, third point. Next one is actually just double checking that all phenols are made and tested. All of mine, I know personally that have already been shipped and tested and is there. So I think that's good on my part. Not sure, Dimitri, do you have any perhaps anything that was
So
missing?
we finished working on Series 1 phenomenals more than 1 year ago, and we currently have all data points from UCL, all the compounds that have been tested on behalf of UCL part. All data points are in the master list because we did not do any additional compounds.
Okay.
Yeah, it was also just to making sure that all the compounds that have been made and tested are of interest are actually in the manuscript, right? Because the first draft of this manuscript was some time ago. Just want to make sure that everything that we think is of interest in the series has made it to the manuscript and we haven't left anything out.
But we decided that like not all of the compounds that were tested would go in the manuscript. So
There was
some-
there was a time when we just sort of like we say this is the cutoff and then we don't add anything new in, and I think that was, I believe, at the end of 2022. So there's some later one would not have been included, but all of the one up to that point that we agreed that it should be in the manuscript is already in the manuscript. Yes.
All right, great. And just provided there's nothing more recent that has been tested, which is of interest, which should go in, right? So certainly not every molecule, but just to make sure that since we first drafted the manuscript, that we haven't got another compound which should be in there, which isn't because we've forgotten about it.
To the best of my knowledge, that should be everything. When I go back to do like the other things for the manuscript, I'll have one final look over my part to see if anything's missing. But I think most of it should already be in the manuscript because I don't recall anything else interesting that was abandoned and unloved. So we say.
Just
So
to
that means
confirm,
that I can make.
Sorry.
So that means that I can make an Excel copy of the data sheet, removing all the other series in that copy, only copy 1 with all the in vitro data and in vivo data. And is it easier for you if I make that prior to when you're checking if all the compounds are there?
I think that
that'd be good.
should be, yeah.
Yeah, that would be a good idea.
All right.
So we're looking for compounds which would still be addressing the SAR gap potentially from the ones that have been synthesized. And if that's the case, then we need to add them to the manuscript. Is that what we're looking for, right? Okay.
Pretty much, yeah.
Yeah.
All right. So next up is actually on series 2. So particularly Rowan's contribution for compounds from Griffith University.
I got an email from him just now. Apparently, Wendy, he's waiting for a shipping address from you. He needs to know where to send the compounds to you, Wendy. That's all. And then he's got them ready to go to ship to your lab. He just needs to know where to send them.
Okay, it is on GitHub, I assume.
Probably, yeah. Yeah.
Okay, I will
somewhere.
check.
Yeah.
And otherwise you
Apparently
can forward the email and then I will put it on the email and put it on GitHub.
I'll do that now. Yeah.
Yeah. So is that for Wendy's address? Is that right? Or is that okay? Next one is crowdsourcing updates from various places.
Yeah,
Okay.
so I think Kimberly's been in touch with all the relevant parties there, right? I think that was the last meeting that they had. So I think she was talking directly to the different schools that were taking part. I haven't caught up with the schools. There are now Sevenoaks, Whitgift, and another one. And there's also a team at the University of York who are currently working on that series. So there's 4 different teams. But I think that Kimberly was the last one to speak with them, as far as I remember.
Okay. So shall we just tag that to Kimberly for an update? Is that all right?
yeah, and also I think that the York team, so Julia, is likely to have been the busiest. So maybe we could try and request an update from her. And there was also someone called Katie Terrett at Barker College, I think, who was working on it, who was working more closely with Kimberly. Maybe she could provide an update as to if they've made a molecule.
Yeah, and potentially this is still in the early stages, but in so like June, July, then Breaking Good will again have more compounds from series 2 from high school students that we work with as well. But that's sort of like a few more months down the track. But yeah, that's kind of like in the work right now. So I think either myself or Clementine should be able to do an update on that when that arrives. I guess also another one on series 2 is a meeting is needed to be had so that we can know like all the targets. Is that done, or do we need to do something about that one?
Sorry, what was the action story? Is that to make sure that every school has a different target? I'm pretty sure that Kimberly did that. I'm pretty sure that was the subject of that meeting. Okay, sounds good. So I'll just take that one off as well. Well, provided that's what happened, right? So okay. if a school is still waiting on a target to be finalized, then we should try and help them do that. Okay, let's just tag that to Kimberly and just double check. Yeah, I'm not too sure about that part. Okay, so next one is on series 5. So oxazoles are quite potent in vivo as well. So we just want to know that if there are more oxazoles out there. So this is where I guess because as I just mentioned, so two honor students from UCIT would like to work on series 5 as well. So firstly, if we have any potent or any oxazoles of interest out there, then that would be great. But also like potentially we can also make some more so as 2 honors projects incoming as well, if that's where we can get some more compounds. But is there any more elaboration on the oxazoles part? Yeah, Bernard, did you manage to reach the NDI about that?
Yes, I'm happy to say that Andrea from Otago and Charlotte from ICL are preparing more compounds at the moment as part of a different project for DNDi, but they agreed to share some mix for Wendy for mycetoma testing then later this year in spring or summer, early summer latest. So it's in progress.
Also, one thing that I would be interested in having as well is if there is a reaction route potentially for one of these series 5, I would be quite interested in seeing, because I did have one where I found off the literature, but I was having troubles with some of the steps. So I would like to, if there's a resource somewhere on that, just so I can double check and perhaps hand to the honor students to have a look at as well, that would be greatly appreciated.
Well, I think Andrea has published a paper about benzoxazoles last year, together with DNDi and other collaborators of that project. And maybe there's some section about the chemistry part they have done together with students, with pupils from their schools.
Okay, yeah, that sounds good. I'll have a look, and if I can have
we can reach out to her, of course, and just ask if the route that they're using is the published one. Okay, sounds good. So I believe like when I was looking at it, it's probably before the time that was published. It was quite early on in the previous year, so I might have caught that one in particular. But yeah, I'll have. Bernard, did you get a sense that there were, because these are samples that are being made, are there any that have come from previous projects that might be sitting in a fridge somewhere? Or did they say no, that we've seen them all?
Well, they sent. I think these compounds they sent were from an older project about leishmaniasis, and it was just repurposed then because of their similarity with the benzimidazoles to have some variations in the heterocycle rings. And fortunately, we found, or Brandy found, that there are some quite active ones among them. And now we must see if they're also active in vivo.
Just wonder if
Yeah,
there are
just
any
to
other...
clarify, we did test them last year in vitro, but we didn't get around in the larvae season to test them in vivo, and that's the plan for this larvae season, to see if those are as promising as the other ones. Ma also constructed a Falciformispora larvae model now, where we get Falciformispora grains like they do in human. The grains look a little bit different like those from Madurella mycetomatis, so it would be good to check if these are active against both.
That's great. I was wondering if, because oxazoles is such a common motif, I just wondered if there may be other supply, compounds around that we could evaluate without having to make them. And so I guess if we took some of the ones that were active as just exemplars and we did a SciFinder search of a similarity-based search, and I wonder if there were some papers that we could have a look at, published in the last few years by some teams that we could then approach to see if they have any samples. It's always quicker to try and get contributions of samples that are already made than trying to make them. I haven't done that search. I don't know if anyone else has done that search as to what the surrounding chemical landscape looks like.
I haven't done it myself. I might have done a little bit just to look at the reaction rate when I first tried it. But I guess that could be something that I also recommended to our newest recruits into the project to have a look at. Might be worth a lit search practice for them to do. I can recommend that to them as well. All right, so next up on the agenda, so Kidoxime, Demetrius already designed some sets of compounds. Would you like to do a brief update on that? We have only like 7 minutes, so.
Yeah, I'll just share the screen quickly. So the brief update on the Series 6 phenomals. So currently, 20 molecules are being prepared for the dispatch, which will happen somewhere around like this week, the latest at the beginning of next week. Just to summarize, there will be 20 molecules in total currently in the upcoming batch, but per each entry there will be 2 molecules to test in the assay, because there are several options of how that might happen. So option one isomeric form plus EZ form. Option B, 2 separated forms, EZ, in cases where that's possible, or where it was not possible to separate the isomers, they will be tested as EZ mixture, as in round 1 biological evaluation of the project for the first part that was done some time ago. The highlighted ones, which were tested previously as EZ mixtures with different scaffolds. So at the moment, because I started to assemble the dispatch package already, I would probably say 16 are ready to go, and 4 are either being synthesized and need characterization and a separation of isomers, or there are 1 or 2. I guess there's 1 substrate which I'm still waiting for from the supplier to run the reaction. But the decision has been made when we think that it's better if I dispatch, divide this package like into 2 parts, I guess, like send what I've got now, and the rest will be sent in the nearest future. I guess that's probably the best option. The list currently adds up to 30 compounds for the Kidoxime Series 6 in total. But I would expect it to be a minimum at least like 35 compounds, because I still look at the SAR gaps, and there are still a few compounds that I will definitely add in my PhD to this list. With the scaffold, that's quite important with the scaffold. Otherwise, we'll be working probably on something new for the rest of the project, for the rest of the time that I've got. So that's everything I have to say on
Okay.
Series 6.
Yeah, thanks. Since we're also on that as well, I just might give a quick update on what I will be planning to do as my final batch as well, which is, sorry, just 1 second.
I'm sorry, there's one thing I forgot to tell.
Oh yeah, sure.
Also on the Keroxam Series 6, because the purification is quite difficult in most cases. So I mainly ended up with E form. So the batch would look probably in most cases as E isomer plus EZ mixture, which is not a problem, which is a way around like going this. So if E is inactive, then EZ, if E is inactive and EZ of the same entry is active, it means that the Z is giving some activity, which might be useful. But what I was thinking about, and as part of the biological, as part of the stability testing, I was thinking, how about like I make the other isomeric form as a salt formulation. So the concept of acid catalysis, I found like in patents, when you manipulate the equilibrium to one form and then you get predominantly one isomeric form at the end of this manipulation. So I was thinking this would be as an option for the missing isomeric form for each entry. Any thoughts, any suggestions on this? Bernard, Matt, what do you think?
So maybe plan what you're thinking and sketch it out so we can take a look. I think we're just running a little low on time.
Yeah.
Sketch it out, put it on GitHub.
Yeah,
No, but it's quite
GitHub.
simple. It's just the salt formulation. The concept is very simple. So I can make the missing isomeric form by using a specific protocol. That's all what I'm asking about. Do you think it's useful?
Salt versus regular, I think it's a separate question from the isomeric question. Salts are always fine, always useful. As a way of purifying one isomer rather than another, that's a selective crystallization. And in that case, often the salt, the counterion that you use isn't the best one to use because often it's got a bulky counterion. Sometimes it's has stereochemical information in itself in order to make a selective crystallization work. And in that case, sometimes the salt isn't the counterion that you normally use for that isn't necessarily the best one for a biological assay.
But the only currently marketed drug, oxyconazole, is a nitrate formulation. That's why I thought this idea would be quite useful to implement, because this is commercially purchased and will be sent to the batch for testing as well. But the concept is usually salt formulations have better pharmacokinetic properties, so that might be useful for grain penetration. I thought like developing this concept might be useful as well. So two things addressing getting the missing isomeric form and also seeing whether that gives like better outcomes in terms of potency and assay results.
Yeah, I think that sounds reasonable to me. I guess
yeah,
we're almost running out of time, so if I can just have 2 minutes, I will also show what I was gonna do as like the last thing for my synthetic part here. So what I'm going to do at the end as well is mainly addressing one of the SAR gap that we are currently having for Phenaramo, and that's the extension of the exploration of the four substitution extension. And one way to do that is actually by Suzuki. And I think Matt has made some suggestion when I put on GitHub as well in terms of like also be mindful of just lowering log D value. So for example, with a amination, CN cross coupling. So that's something that I will be exploring a bit later, but I also have some route on Suzuki coupling. So Marat has done a lot of this work here. I did find from his thesis the reaction route with Suzuki couplings using this palladium complexing as well. So I was thinking I'm just going to use some of that to make this extension of the phenol with a carboxylic acid, because it's also quite its log is quite low. When I double check that, so I can make a small batch of this one. And I also go on to do a few coupling using the Bushveld Hardwick to make a few more so like this motif as well. But this is currently in planning, so I don't have a particular target yet. So what I'm thinking is that because I can do this, and if I can do this well, I'll just get some of my final compounds that I have, like in the 100 milligrams of, and I just quickly do a late state functionalization to get some more compound out for testing. So that's the most efficient route to getting more of these out. And in the meantime, while I was looking at this, I can also do address another SAR gap, which is actually the match pairing in the change of the pyridyl ring itself. So like with these kind of motif here as well. So I'm doing this while I'm looking for a way to make this work, and then hopefully I'll get both out at the end. So this one here in particular will actually address, just add into the missing bits in here as well as that one right there. So it will just bulk this section out a little bit more, and I will be making that personally. So yeah, that's kind of my plan, very briefly.
Hung, if you need, you can have a look at the lab archives, because I did Buchwald Hardwick for one of the scaffolds in the series 1 Panoramos. I think it was microwave synthesis, but we can double check with this one we need.
Oh yeah, I think
you need the
Shirley actually mentioned that as well. I would love to have a look at your reaction condition and so see if it's actually applicable to this as well.
Sure.
Yeah, if I can't find, I'll GitHub you for help. Okay. Yeah. Yeah, that's all from me. I think we are slightly over time, 3 minutes. Is there anything else that that is quite urgent that needs attention before I?
Sorry to leave before the Erasmus update, but I've just got to nip to a budget meeting, which I can't miss. So I'm gonna have to say bye.
Okay,
right.
no, see you. Thanks for coming. Is there anything else that we need to like address right now instead of a bit later?
No, we are at Erasmus are still busy screening. We have a lot of compounds to be screened, so it might take a little bit longer for us to update things on GitHub. But we are, especially Ma is very hard working to get everything done.
Thanks, Ma. Thanks for your work. I'm also doing a little bit of, I'm also doing a little bit of writing as well, so I'm not quite fast on the getting compounds to ship, but I hopefully will get that out in probably 4 or 5 weeks for one more batch.
I'm definitely sending my compounds this week or the beginning of next, because most of them are complete and it's just like the missing bits, but I think it's not worth wasting time. So I'll just send them like in 2 different parts, as I said previously.
Okay.
Wally, do you have anything else which you would like to add? Also, thank you for coming to the meeting as well. Lovely to see you. I think it's my first time seeing you as well.
Yeah, this is certainly my first time here. Thank you for having me. Very, very exciting to see all the work that's being conducted. I would love to contribute in any way possible, in particular in the form of any type of bioinformatic discovery or computations that you all need to perform. I would say I'm quite versed and adept at structural prediction, calculating free binding energy, and a number of other drug discovery related bioinformatic tools. So yeah, please do reach out to me if there's anything I can do to help, or if you guys have any ideas. And I did have one really quick question. Does anybody here use Schrodinger or Live Design, or have you heard of it?
No, most of my bioinformatical work so far has been together with Imad Abukassaya from Recon. Usually I just ask him if something is needed, but yeah, I don't do it. I'm a biologist, so I pipette in the lab.
Gotcha.
I have very limited computational chemistry knowledge or computational biology knowledge. So like I would be like any help from you would be greatly appreciated. Like I'm not quite well versed in that area, unfortunately. I did pass my quantum chemistry tests for undergrad.
I think you actually fill one of our gaps here. Your expertise.
Amazing.
We
really.
definitely need computational help with the analysis of the target molecule.
Yeah, that's like
Absolutely.
one area that we kind of miss, and we are really hoping someone could help fill that gap. So I think you would be a great fit for our team in the coming.
Awesome. That's really great to hear. I'm very well versed in genomic data analysis and computational chemistry, and I work with a world-class team of computational chemists. So if there's anything that's outside of my wheelhouse, they're always willing to answer any questions for me. I have a really great relationship with everybody that I work with. So please do reach out. I'm going to leave my email address here, and I will follow up directly with Wendy and Matt.
Yeah, so thank you so much, everyone, for coming. I think we're just 7 minutes over, but so I'm just gonna call it meetings adjourned for now. But love to see you, and I hope to see you again soon. And if there's anything, obviously, just put it on GitHub, and we'll try our best to respond in a timely manner.
Awesome.
All right. So
Thanks everyone for coming.
Thanks everyone.
Thanks everyone. Take care.
Bye.
Bye-bye.
I'll message me when you have time, right? I can adjust.
Okay. Yes, I'll put it on GitHub, and we'll
I'm
work.
quite flexible, so it's not a problem. But I think it's gonna be better if we just do it together and much quicker.
Okay. Yeah, sounds good.
Yep. See you. Good night.
Yeah, see ya.